Hello everyone and welcome to a recorded lecture of BIO108 online. Today we'll be discussing chapter 13, which is on the lymphatic system and immunity. So the lymphoid or lymphatic system is a collection of lymphatic vessels and lymph nodes found throughout the body. We also have some organs associated with the lymph system that include the spleen, the thymus, and bone marrow. We also have some lymphoid tissues associated with mucous membranes. So we'll be discussing all of these aspects of the lymphatic system. The functions of the lymphatic system are to balance the fluid between blood and tissues. We want to make sure that there's not too much accumulation of fluid in our tissues. So if there is an accumulation of fluid in our tissues, that fluid could then go to the lymph vessels, which can drain that fluid. Lymphatic vessels also help absorb lipids during digestion, and lymphatic vessels in our intestines specifically can absorb lipids that we eat. And the main function we'll be talking about today is the immune function of the lymphoid system that protects us from disease. So the lymph system and the immune system are very closely related. The immune system is just a collection of cells and cell products found throughout the body. It's not like there's just one place for the immune system. Right? It's found throughout the body. And largely, a lot of the immune responses would occur in the lymphatic system. The immune system is responsible for sensing and responding to the microbial environment of our body, and it prevents us from getting infected by a pathogen. So the immune system fights against infections. It also helps us repair injuries. And like I mentioned, many cells of the immune system live in the lymphoid system or pass through the lymphoid organs on their way to other tissues. So that's why they're so closely related. Right? Immune responses often happen in the lymphoid system. So the lymphoid system is closely related to the cardiovascular system that we spoke about just recently. And it runs in parallel to blood vessels. So the lymph system runs in parallel to blood vessels. And the lymph vessels don't contain blood, but they contain a clear fluid called lymph. So lymph is the fluid inside of lymphatic vessels. Within that fluid, there are also immune cells that we'll talk about, called lymphocytes, that can find debris or bacteria and gobble them up before they have a chance to affect us. There's also lots of proteins found in lymph. So what is this lymph all about? Well, we spoke about last time that blood capillaries leak excess plasma, excess fluid into tissues. So if this is our tissue, these are the cells of the tissue, and this is the fluid outside of the tissue. When there's too much fluid in the blood, that excess fluid goes in the tissue. Well, we don't want that fluid to accumulate in the tissue. So where does it go? It actually gets drained in these closed-ended lymphatic capillaries. So excess fluid from the tissue, from the interstitial fluid of the tissue, can be drained through these lymphatic capillaries. This is a prevention against edema. Edema is swelling. So if we didn't have lymphatic vessels, we wouldn't have a way of draining any excess fluid in the tissues. And remember that fluid got put there from our capillaries, from the cardiovascular system. So the lymphatic vessels drain excess interstitial fluid and returns it to the blood in a one-way flow. So it's a one-way street. So the fluid will go back from the tissue to the lymph, back into the blood. Right? The fluid is eventually returned back into the blood circulation. Right? And remember that it's always a one-way street. So fluid is coming only from the tissue into lymph. It doesn't go from the lymph to the tissue. So that's something that you should keep in mind. Once the fluid enters lymphatic capillaries, they enter lymphatic vessels that lead to lymphatic ducts. And the lymphatic ducts are emptied into large veins near the heart. So again, all that fluid enters the blood supply through the subclavian veins. The way that the fluid can travel through these vessels is very similar to how blood travels through veins. There are these valves, right, that are one way that basically make sure fluid can only go toward this vein. It's not going to go backward, right? And it's going to only go toward this vein, right, through the ducts. So there are little valves that once the fluid gets to this point, this valve will close and prevent it from going backwards. Very much like the way that the veins allow the blood to be returned to the heart. So here we have a nice picture of the lymphoid system, which shows the several types of interconnected organs. So the lymphoid organs. And this shows the nice, the circuitry of the lymphatic system, right? It shows all the different pathways. And specifically, lymph from the right upper limb, the right side of the head and neck, and the right thoracic region all drains through the right lymphatic duct, which is shown here. So all these, look at all these vessels and how they all drain into this duct over here. And then they drain into what we call the right subclavian vein. Lymph from the remainder of the body, so everywhere else you can see lymph vessels, all drain into what we call the thoracic duct. This is the thoracic duct. And the thoracic duct drains into the left subclavian vein. So I just want to go back 1 second when I show you. So here we have the first organ we're going to talk about, the thymus. So the thymus is one of the primary lymphoid organs in addition to red bone marrow. So primary lymphoid organs are called primary because lymphocytes originate there. So lymphocytes originate in primary lymphoid organs like the bone marrow, the red bone marrow, and the thymus. All white blood cells originate or mature in these organs. Lymphocytes are white blood cells that are critical for the immune system. And like their name suggests, they originate in lymphoid organs. They're lymphocytes. Specifically, if the lymphocytes can recognize pathogens and activate the immune system, we say that they're immunocompetent. So not all lymphocytes formed in the red bone marrow are immunocompetent. To be immunocompetent, they have to be able to elicit an immune response. So they have to do a little bit of maturation before they can be immunocompetent. And I'll talk about that in a bit. We also have secondary lymphoid organs, which is where the lymphocytes and other immune cells go. And that's where they can basically recognize a foreign invader and initiate an immune response. So lymphocytes originate in the red bone marrow and thymus. They then go to the lymph nodes and the spleen, where they can do what they need to do. And they're immunocompetent because they can actually recognize and attack a foreign substance. So we said red bone marrow is one of the primary lymphoid organs. And where is red bone marrow located? Well, we know that it's in the spongy bone of the skeleton, right, in the spongy bone. So we have all of our blood cells originate in our red bone marrow, including the immune cells. So we're going to talk about 3 different kinds of lymphocytes. We'll talk about the 2 main ones, B cells and T cells, B lymphocytes and T lymphocytes. B lymphocytes originate and mature in the bone marrow. You can remember B for bone marrow. Then they migrate throughout the blood and the lymphatic system. So there are B cells or B lymphocytes stay in the bone marrow, until they are immunocompetent, and then they can move on to their secondary lymphoid organs. T cells originate in the bone marrow as well, but then they have to move to the thymus for maturation. Then they can go to their secondary lymphoid organs. So in this picture, we know all hemocytoblasts are the blood stem cells. They can then differentiate into pre-B cells or pre-T cells. Those pre-B cells can then mature in the red bone marrow and then migrate to the lymph nodes and the spleen. However, T cells never mature fully in the bone marrow. They have to go to the thymus before they can then be immunocompetent, and then they can go throughout the lymphatic vessels and into the lymph nodes and spleen to do their job. So this is a picture from the book. We have undifferentiated lymphocytes being produced in the red bone marrow. They could then go to the thymus and become immunocompetent T cells, and then they can go do their T cell thing that we'll talk about. Or others will stay in the bone marrow to become immunocompetent B cells, and then they're going to be transported through the blood to do their B cell thing, right? They're going to go through the lymph nodes and the spleen, etc. So let's talk about the thymus. So the thymus is a glandular lymphoid organ in the thoracic cavity, just superior to the heart. It's located in the mediastinum cavity. It's actually really large in infants and children, but after puberty, it becomes very small and starts losing a lot of the thymus cells and becomes a lot more connective tissue and fat. The functions of the thymus are to differentiate T cells into becoming immunocompetent. So they produce thymosins, these hormones that promote the differentiation and the division of T cells that can make them immunocompetent. So without thymosins, we would not have immunocompetent T cells. And after they're mature, those T cells can then be distributed to the blood, to the spleen and lymph nodes. Also, there are other lymphoid tissues throughout the body, not just spleen and lymph nodes. But we'll talk about that in a bit. So for now, I'd like you to pause here and answer this question. So the answer to this question is A, the red bone marrow. Where do they mature? Pause here. The answer is C, the thymus. Where do B cells originate? The answer is A, the red bone marrow. And where do they mature? So pause here. And the answer is A, also the red bone marrow. Great. So let's talk about lymph nodes. Lymph nodes are secondary lymphoid organs that filter lymph. They're roughly bean-shaped. This is one big lymph node. This is a lymph node. And they're found throughout the body, but they're usually found in groups along larger lymphatic vessels. We have large clumps of them. So let me just show you. These lymph vessels are the lines, and these bean-like things are lymph nodes. And they occur in clusters. So here we have a cluster of axillary lymph nodes by the armpit. So lymph nodes are sites of immune system reactions. They receive both blood supply and lymph supply. So all of those cells, or anything that's in the blood that shouldn't be there, or anything in the lymph that shouldn't be there, gets filtered and cleansed by our lymph nodes. Right? So they go in one way and they come out another. And inside these lymph nodes, what does the cleansing are macrophages and lymphocytes. Lymphocytes, we just mentioned, B and T cells can initiate an immune response. We'll talk about how that happens in a bit. And macrophages can use phagocytosis to engulf any infected cells or any debris and chop them up and destroy them. So macrophages are cells of the immune system that could eat other cells and digest them. So they live inside of these lymph nodes throughout our body. So as our blood and lymph gets filtered through or gets sent through here, it gets filtered out, right? All the bad stuff gets eaten up and destroyed by macrophages, and possibly an immune response could be initiated by lymphocytes. And again, like I mentioned, they're found clustered specific throughout the body, but specifically we have clumps in our neck, by our thoracic, or by the collarbone, and our armpits, and also inguinal by the groin. All right, so we said lymph nodes filter and cleanse the lymph. That means bacteria, viruses, any cellular debris, also any abnormal cells will be trapped in lymph nodes. Those macrophages would then engulf and digest those foreign materials, so they can't make it any further into our body. Lymphocytes will recognize infections, will recognize, oh, this is bad, and then can activate an immune response. There are no lymph nodes in the central nervous system, interestingly, but they're found all throughout the body elsewhere. And again, we can see, right, the really wide distribution of lymph nodes. So look at all those beans, right? And within those little beans, here we have a close-up, right? We have B cells and T cells and macrophages. So they're the cells of the immune system that are going to make sure that we're constantly filtering out anything that shouldn't be in our blood, in our lymphatic system. I also just want to go back and mention that within each lymph node, you have lymph nodules, which are these smaller little clumps. And we'll talk a little bit more about the lymphoid nodules, but that's specifically where the lymphocytes and macrophages kind of reside. So here's a lymphoid nodule, and I'm going to mention this in a bit. So just remember that within a lymph node, you have lymphoid nodules, which has the macrophages and the lymphocytes. So the spleen is the largest lymphoid organ, the spleen. So it's a secondary lymphoid organ because it can't make its own lymphocytes. So we call it a secondary lymphoid organ. It's located just behind the stomach, under the diaphragm, in the left upper quadrant of the abdominal cavity. And it's basically like a giant lymph node in structure. It has a lot of different compartments in there. We're not going to talk too much about that. But the tissue has two different functions, and we'll talk about that soon. So it receives both blood and lymph, and it will filter bacteria, any damaged cells, any cellular debris from both our blood supply and our lymph supply. So just like a lymph node filters out blood and lymph, spleen filters out blood and lymph. It also has tons of lymphocytes. It doesn't make the lymphocytes, but lymphocytes reside there. Specifically, the lymphocytes are found in the white pulp. And this is where a lot of the immune system activation happens. There's also a circulatory role of the spleen. If you remember, it removes and recycles old or damaged red blood cells from our blood supply. It's also a storage area for red blood cells and platelets. So the spleen has a role in the circulatory system, the cardiovascular system, also an immune system role. Interestingly, a person could survive without a spleen. It's not essential to life, but following a splenectomy, you may become more susceptible to a bacterial or viral infection. So without a spleen, the functions also happen in lymph nodes. They can also happen in the liver, but you can still be more susceptible to a pathogenic infection. So pause here and try to answer this question. And the answer is B, is the best answer. So answer this question and pause. And the answer is A, the spleen. Let's talk about other lymph structures. So we have tonsils, which are in our mouth and pharynx, and our throat, that just like all lymphoid tissue, they can help protect us against pathogens. They contain lymphocytes and macrophages. Right? And the lymphocytes, again, recognize pathogens. Macrophages consume foreign materials or any infected cells. We have 3 types of tonsils. We're not going to talk too much about those. But they also include the adenoids, which are the pharyngeal tonsils. Lacteals we'll talk a lot more about during the digestive system, but lacteals in the small intestine absorb lipids from digestion into the lymph, and then they go into the bloodstream. So we'll talk about lacteals in the small intestine. They have lymph in them, so it's part of the lymphatic system. It drains into the lymphatic system. MALT are mucosa-associated lymphoid tissues. So if you recall, remember those lymphoid nodules that are found in the lymph nodes? Well, we have those nodules all over our mucous membranes. So we have these nodules that have macrophages and lymphocytes trapped in special tissue to provide additional barriers to invasion by pathogens. We have large clusters of these malts in our respiratory, digestive, urinary, and reproductive tracts. So we have these nodules all over our body, wherever there is a mucosa. Remember, we have mucous membranes lining any part of the body that's exposed to the outside environment. So we have mucous membranes, and in those mucous membranes, in the areolar tissue, we have these lymphoid nodules, these little clumps of macrophages and lymphocytes that can help fight against infection. Right? So that's if we're exposed to the outside and something foreign can get inside of us, we luckily have lymphocytes and macrophages to do the fighting for us. The appendix is part of the mucosa-associated lymphoid tissues that control bacterial growth in the large intestine. And the appendix could actually be removed without any serious health risk. It used to be known as a vestigial organ, an organ that was only needed by our foreign ancestors, like other mammals. But we know that it does have a role. It does help control bacterial growth. So we are thankful for our appendix. So this is a great table to stop at. Make sure you understand each of the components of the lymphoid system, how it's characterized, and then what its function is. So let's watch this video together.
The lymphatic system is a network of vessels, lymphatic tissues, and organs, and lymphatic fluid known as lymph. As a system, it responds to pathogens and foreign substances. Throughout the body, fluid similar to blood plasma seeps from the blood capillaries into the spaces between cells. Approximately 10% of this fluid enters blind-ended lymphatic capillaries and is known as lymph. Lymphatic capillaries are microscopic vessels that are found among most blood capillary networks. Lymphatic capillaries converge to form lymphatic collecting vessels that merge to form lymphatic trunks. Lymphatic trunks drain into the largest lymphatic vessels, the right lymphatic duct and the thoracic duct. The right lymphatic duct receives lymph from the right upper limb, thorax, neck, and head. It empties into the right subclavian vein. The thoracic duct is larger than the right lymphatic duct and receives lymph from the remaining regions of the body. It empties into the left subclavian vein. Lymphatic tissues include nodules and organs. Lymphatic nodules are collections of lymphocytes that lack the connective tissue capsule. They contain B lymphocytes, T lymphocytes, and macrophages that attack pathogens. Clusters of lymphatic nodules are associated with the walls of the gastrointestinal, respiratory, urinary, and reproductive tracts. Tonsils are large groups of lymphatic nodules found in the walls of the nasal and oral cavities. Tonsils provide protection against bacteria and other harmful materials that may enter the nasal or oral cavities. Pharyngeal tonsils are located in the nasopharynx and when inflamed are known as adenoids. Palatine tonsils, commonly referred to as the tonsils, are located at the posterolateral aspect of the oral cavity. Lingual tonsils are found at the root of the tongue. Lymphatic organs, including lymph nodes, thymus, and spleen, consist of clusters of lymphatic cells that are surrounded by a connective tissue capsule. Lymph nodes are small, bean-shaped structures located along the course of lymphatic vessels. In selected regions of the body, lymph nodes are found in prominent clusters. These are particularly evident in the axilla or armpit, the inguinal region or groin, and in the cervical or neck region. The primary functions of the lymph node are the production of new lymphocytes, filtration of the lymph, and initiation of an immune response when necessary. A lymph node consists of an outer cortex, which contains lymphatic nodules, and an inner medulla that contains connective tissue cords lined by lymphatic cells. Lymph enters the node through afferent lymphatic vessels and courses through channels called lymphatic sinuses in both the cortex and medulla. After filtration, lymph leaves the opposite side of the node through efferent lymphatic vessels. The thymus is located in the thorax, anterior and superior to the heart. It is the site of formation and maturation of T lymphocytes and is most active during childhood. Starting in adolescence, the thymus is progressively replaced by fat and connective tissue and eventually becomes non-functional. The spleen is the body's largest lymphatic organ. It lies in the abdominal cavity, just inferior to the diaphragm on the left side of the body. The spleen filters the blood, removing old or defective blood cells, foreign materials, and pathogens. It also helps to regulate circulating blood volume. Like lymph nodes, the spleen is involved in the production of new lymphocytes and in initiating immune responses.
Great. Now let's start talking a little bit more about immunity. So I'd like you to pause here and watch this video, watch this YouTube link, just to get a good orientation about how our immune system works. I think that'll help a lot moving forward. So why do we need an immune system? Well, we need to protect ourselves against diseases. But what causes diseases? Pathogens do. Pathogens are microscopic, infectious substances like viruses, bacteria, parasites, and fungi. For example, a virus is composed of DNA or RNA surrounded by a protein coat. And the way that a virus works is that they insert their genes into a host cell, and they force the host cell to make viral proteins so they can replicate. So the viral RNA uses the host cell to create new RNA and proteins, so then more viruses could be assembled and they can burst out of the cell and then infect other host cells. Are viruses alive? Right? They can replicate, they have biomolecules, they have DNA or RNA, they have proteins. So are they alive? They're not alive, right? Because they're not made of cells and they're not capable of reproduction on their own, as well as a couple of other factors. Viruses are not alive. They require other cells to propagate themselves. And not only animal cells get infected by viruses, plants get infected by viruses, bacteria even get infected by viruses. So a bacteriophage is a virus that infects bacteria. They infect bacteria or infect a bacterium. So here's a bacterium over here that is getting infected by a bacteriophage. And this is the nucleic acid over here that's going to be inserted into the bacterial cell. And the bacterial cell will then make more bacteriophage. And the viruses will then burst from the bacterial cell and then infect other bacteria. So here is a bacterium. And bacteria are single-celled organisms that are mostly harmless. They could actually be beneficial sometimes, and in rare cases, they could be harmful. So bacteria have a bad reputation for being harmful, but most bacteria are harmless. They have no effect on us that we know about. Many can be beneficial, few can be harmful. You are actually made up of more bacterial cells than human cells. Because you have so many bacteria in your gut and they're so much smaller than human cells, you are made of more bacteria. You have more bacterial cells in you than human cells. The ones that are harmful are few and far between, but we know a lot more about them. And they're harmful because they release toxins or harmful enzymes. So some harmful bacteria would include Streptococcus that cause strep throat, or Salmonella that cause food poisoning. Some viruses would include influenza or rhinovirus, or we know a lot about SARS-CoV-2 is the strain that causes COVID. All fungi can cause athlete's foot, or some parasites like Plasmodium can cause malaria. These are all different diseases that are controlled by our immune system, right? Our immune system is responsible for making sure that any of these microbes, any of these pathogens cannot cause disease. So there are two divisions of the immune system. First, we'll discuss nonspecific immunity. And nonspecific immunity or nonspecific resistance provides protection against all pathogens and foreign substances, but it is not directed against a specific pathogen. So these include mechanical barriers and the factory settings of the immune cells. So basically, the mechanical barriers of our body that make it very hard for pathogens to enter, they're just nonspecific. It's general to any pathogen. And also we have immune cells that just kill any cell that's infected, right? They're not against a specific type of pathogen. So these are like the general police guard, right? If something's going wrong, the nonspecific response will take care of it. If it's, again, but it's not against a particular pathogen. Adaptive immunity is specific. So in adaptive immunity, we're going to see that lymphocytes learn and remember specific pathogens, and they produce a targeted response. So it's a specific response to a specific pathogen. This is like the SWAT team, right? These are trained cells that are looking for one specific pathogen and know exactly how to get to that pathogen. So they're a lot more trained. We'll talk about nonspecific resistance first. Right, nonspecific resistance includes mechanical barriers, some chemical actions, phagocytosis, inflammation, fever, and also we'll talk about these other lymphocytes called natural killer cells. But let's talk about mechanical barriers first. The most obvious mechanical barriers against pathogens are the skin and mucous membranes. So think about the skin. Skin has tightly packed epidermal cells. It's very hard for a pathogen to crawl in between the spaces of epidermal cells and get inside of our bloodstream. Mucous membranes have very sticky mucus that are very good at trapping any pathogens or airborne particles and preventing them from contacting the underlying membrane. So the idea of mucus is any pathogen that's inhaled or ingested can be trapped and destroyed by the tonsils before it gets inside of the body. Tears and saliva and also urine, also mechanically, right? They're physically washing away pathogens that could enter through the eyes, the mouth, or the urethra. We also have chemical actions. So various body chemicals like enzymes can actually defend against pathogens. But this is non-specific. It's not against a specific pathogen. They just do it toward any pathogen or any bacteria, for example. So lysozyme is an enzyme. Lysozyme. Lyso means to cut. So lysozyme is an enzyme that cuts open bacterial cell walls. And lysozyme is found in our tears and our saliva and our nasal secretions. So just non-specifically, we have, like, that's where Lysol comes from, by the way. So Lysol kills bacteria. Lysozyme is the natural Lysol that's just found in our secretions. Here's a picture of lysozyme. Gastric juice is very, very, very acidic. It has a very low pH. That will prevent any bacteria from being able to survive the stomach, or many bacteria from being able to survive the stomach. Also, there is an enzyme called pepsin. And pepsin is an enzyme that digests any pathogenic protein. The blood contains a group of plasma proteins known as complement. So complement are blood proteins that can attack bacteria. They attack bacteria by physically punching holes in the bacterial cell membranes. Really fascinating. I could talk a lot more about them. But these complement, what you should know right now is that complement proteins punch holes in the plasma membranes, and those plasma membranes will then burst open and the pathogen will be destroyed. Then some macrophages can come in and clean up the mess. Interferon is something that is a chemical that inhibits viral replication in neighboring cells. I'm going to show you a video of interferon. So it's a chemical produced by cells that are infected by a virus that inhibits uninfected cells from getting infected by a virus too. It activates an antiviral mechanism. So what enzyme is found in tear, saliva, and nasal secretions? The answer is C, lysozyme. So here is a quick video on interferon.
When a cell is infected by a virus, the virus enters the cell and produces structures that are not found in uninfected cells. The presence of this viral material signals the cell to produce interferon. The interferon moves out of the cell and attaches to receptors on nearby cells of the same type. The cell that produces the interferon is unable to save itself. The virus replicates in this cell and then moves out to infect nearby cells. The nearby cell that already has interferon bound to its surface responds in several ways, including production of enzymes that degrade messenger RNA and prevent protein synthesis. Thus, a virus can attach and enter the cell, but completion of the viral replication cycle is prevented.
So very cool process there. So another type of non-specific resistance is phagocytosis, or cell eating, which is the engulfing and destruction of pathogens or any type of damaged or cancerous cell or cellular debris. This is carried out by white blood cells, the neutrophils, and monocytes. So neutrophils are the most abundant white blood cell, and these are capable of eating a pathogen, and then it kills itself. So a neutrophil consumes a pathogen and then kills itself. And that's actually what forms pus. That's what neutrophils are. And these neutrophils and monocytes are attracted to an infection. And when monocytes leave the blood and enter infected tissue, they become macrophages. So monocytes and neutrophils go through the blood, and they could then migrate into tissues and then engulf pathogens. So here is a macrophage. Here's a virus outside of the macrophage, and this is physically engulfing this virus by endocytosis, which is called phagocytosis specifically. In the case of a macrophage, macrophages, they engulf and digest the pathogens, and these pathogens fuse with lysosomes, and lysosomes digest all that material, and then it gets discarded as waste. Macrophages can wander among tissues. They kind of search out for pathogens or cellular debris. Others become stationary and are fixed in one place, where they can phagocytize any pathogens that are passing by. So we have a lot of fixed macrophages in the inner walls of blood vessels, in lymph vessels, in the spleen, lymph nodes, red bone marrow, the liver. We need to have constant filtering and consumption of any debris or any kind of pathogen in our body. So we have macrophages all over the place. Let's watch a video of phagocytosis. So they're migrating here. Another type of nonspecific resistance is inflammation. And inflammation is a localized response to infection or injury, and this will promote the destruction of pathogens and promote the healing process. It's characterized by redness, pain, heat, and swelling. So a lot of mechanisms produce chemicals that cause inflammation in response to injury or infection. So it's a chain of chemical signals, and at the end, white blood cells will be attracted to the site of damage. There, the white blood cells can fight the infection, clean up any cellular debris, and fight to repair the injury. So we want short-term inflammation. It's healthy because that will tell all of our white blood cells where to go. It also attracts fluid from blood capillaries that have fibrinogen and fibroblasts. And if you remember, fibrinogen is very important for blood clotting. So we want to make sure that blood clots can seal off the infected area and prevent the spread of pathogens to neighboring tissue. We also have fibroblasts that produce collagen so we can help heal the tissue. So we want this to happen short term in response to infection or injury. Chronic inflammation is abnormal, and this can be a sign of an immune system dysfunction, where you have constant swelling or constant pain throughout the body because you have immune cells thinking that there's something to fight, but there really isn't anything there. Another type of nonspecific resistance is fever, which we all know is a temporary increase in body temperature. And this is pretty important, and again, that it doesn't go too far, right? Because this can kill us if we have too high of a fever. But a short burst of temperature increase can inhibit the growth of certain pathogens, and it also can mildly increase the rate of our immune responses. Our enzymes can work slightly faster at slightly increased temperatures. Finally, the last type of nonspecific resistance we'll talk about are natural killer cells. This is another type of lymphocyte. So we have B lymphocytes, T lymphocytes, and natural killer cells. Those are the three types of lymphocytes. These lymphocytes are not specific. B cells and T cells are specific. Natural killer cells are the lymphocytes that are non-specific. They are normally going around your body right now looking for unhealthy cells. They're just like the suicide, or not suicide squad. I don't know, what do we call them? I guess they're assassins. They're natural assassins that go through the body and they're just killing any cells that are infected by viruses, any cells that is cancerous. They basically just tell the cell to kill itself. And it's doing it non-specifically. So it's not looking for a specific type of virus. They're just doing immune surveillance by traveling through the body looking for unhealthy cells and making sure that they die. So these differentiate and mature in the bone marrow, also the lymph nodes, spleen, tonsils, and thymus. And then they enter into circulation where they go and find any infected cells or cancer cells. Right, so it's pretty cool to think about that we have natural fighters against cancer. Right now, we're always developing mutations that can lead to uncontrolled cell growth. And once those cells do become uncontrolled, we have other cells, the natural killer cells, that recognize them and tell them to die. So we have a natural homeostatic mechanism, right? If our cells grow too much, we have other cells that tell them that they have to die. It's when that fails that we have a tumor form, and tumors that are metastatic could then spread to other parts of the body, and it becomes very hard for our natural immune system to take care of those tumors that are spread throughout the body. And we'll talk more about cancer in another lecture. Now we can talk about specific resistance. Specific resistance is also known as adaptive immunity, and this is where lymphocytes can learn and remember specific pathogens. So immunity is directed at specific antigens. And we'll talk about what an antigen is in a bit. Once lymphocytes have learned to recognize an infection, their response will be swifter and improved the next time the same infection appears. So in order for adaptive immunity to work, we need both immune cells, we need the lymphocytes, and we also need antibodies. So we'll talk about two different types. We'll talk about cell-mediated immunity and antibody-mediated immunity. Both are part of adaptive immunity because both are kind of trained and they remember an infection for a later time. So pathogens are usually distinguished from normal cells by surface antigens. And an antigen is any substance that can cause an immune response. So an antigen is usually a protein or a protein fragment that's on the membrane surface of a bacterium or a virus. You can think about the spike protein as the antigen that your antibodies will recognize after you get the vaccine for COVID. The COVID vaccine is the mRNA code, or I should say the Pfizer and Moderna vaccines are the mRNA code to make the spike protein antigen normally found on the SARS-CoV-2 virus. So pathogens are recognized, antigens are recognized by the immune system and cause an immune response. And an immune response will involve the production of specific cells and substances to attack that antigen. So because this is part of immunity, B and T cells will learn to recognize any antigen and remember it so that the next time that same antigen enters the body, it won't cause an infection, ideally. So B and T cells have to learn how to recognize what is our own cells versus what is a pathogen. And it does this using specific antigens. It can recognize which antigens come from a pathogen, and it can recognize which antigens are self. Each B or T cell can only recognize one specific foreign antigen. So this is really important. Each B and T cell can only recognize one specific foreign antigen. So we need millions, if not billions, of different types of B and T cells to recognize any type of foreign antigen we can encounter in the universe. So we have to have different B cells, each recognizing different parts of unique antigens. For example, cancer cells or cells infected by a virus are recognized by T cells. A T cell would recognize a specific part of a virus. So certain T cells would recognize the spike protein of the SARS-CoV-2 virus. Other T cells will recognize specific cancer cells because of the antigens that cancer cells display on their cell surface. So let's watch a little bit about antigens and antibodies and how they interact. We know a lot more about antibodies after we spoke about blood type, but we'll talk a little bit more about it now.
Antigens are macromolecules, usually of molecular weight greater than 10,000, such as proteins and polysaccharides. They are recognized by the immune system as foreign. Individual antibodies are not made against the entire antigen molecule, but rather to particular chemical groups on the molecules known as antigenic determinants or epitopes. Many different antibodies can be made against a single antigen, each antibody reacting with a different epitope. Complex structures, such as the surfaces of bacterial cells, may have many different epitopes. Each different antibody binds only to the correct epitope. Antigens are...
So it's a little more specific than you need to know, and there's so much to be said about this, of course. But antibodies only recognize one part of an antigen. So one antigen can actually be recognized by many different antibodies. So we have, again, we have millions or billions of different types of lymphocytes that each recognize different parts of pathogens. So specific immunity, we said, is activated by the presence of foreign antigens and then has two branches, right? The immune response will involve one or both of these processes. We have cell-mediated immunity and then antibody-mediated immunity. In cell-mediated immunity, we're going to see T cells play the starring role. So T cells will be activated in cell-mediated immunity, and phagocytosis will be stimulated. This will cause the direct attack against the foreign antigen by chemical or physical means, meaning we're just destroying any diseased cell or any pathogen that's inside of a cell by cutting it apart, cutting it open, or releasing a poison. So cell-mediated immunity is cells are killing infected cells. Right. So T cells are part of cell-mediated immunity. They're responsible for killing cells that are already infected. So they're attacking the invaders that are already inside of the cells. So it's cell-mediated immunity. These are cells that are directly attacking diseased cells. The other way we have specific immunity is with antibodies. And in antibody-mediated immunity, B cells are activated and start producing antibodies against the specific antigen. When that antigen is found, let's say, in the blood or lymph, B cells recognize it and then can bind extracellular pathogens for destruction by phagocytosis or other means. So antibodies can attack invaders that are outside of the cells. Right? These antibodies recognize either proteins that are in or pathogens that are in the bloodstream or that are outside of the cells, whereas T cells attack invaders that are inside of the cells. That's a good way to distinguish between the T cells and the B cells. This is very, very basic, by the way. This is a very broad generalization. And in anatomy and physiology, we'll talk a lot more about how the different T cells do what they do and the B cells do what they do. We're going to talk a bit about each right now. So we'll talk about T cells first. So cell-mediated immunity is carried out through T lymphocytes. And these T cells directly target foreign cells or diseased body cells. They also develop a memory for the antigens in case that they reappear at a later time. So T cells will recognize foreign cells, diseased body cells, such as a transplanted cell or cancer cell, and they'll develop a memory of their antigens if they reappear. There are three main types of T cells. There are cytotoxic T cells that can kill cells that have a foreign antigen. So it kills any cell that has an antigen that makes it a foreign one, makes it not desirable to be in us. And it kills these cells using toxic chemicals. So it basically uses like a poison to kill foreign infected cells. We then have helper T cells that when they're activated, release a chemical called cytokines. And these cytokines activate B cells and cytotoxic T cells. Helper T cells also process and present antigens to stimulate phagocytosis by macrophages. So helper T cells are like an intermediary. They can activate B cells, they can stimulate phagocytosis, and they can also activate B cells. I don't know if I said that. So I'm sorry. They activate B cells, T cells, and they can stimulate phagocytosis. So they're helpers in many ways. They're helping all different kinds of immune cells work together. And finally, memory T cells are dormant cells that are only activated once the antigen re-enters the body after a second time. So they launch a secondary immune response if the antigen re-enters and fits one of their receptors. So how do T cells recognize foreign antigens and then start killing these cells? Well, we have to remember macrophages are the ones that are engulfing the foreign pathogen, and they basically chop up the pathogen into different pieces and they display them on their surface. So a macrophage engulfs the foreign antigen and then displays the antigen on the cell surface. We call these antigen-presenting cells because they're presenting the antigen on their surface. And again, these macrophages are just digesting any pathogen that looks suspicious, chops it up, and displays it on its surface. It's displaying it for the T cells to see. Now remember, there's only one type of T cell out of all the millions in your immune system that will recognize this type of fragment of this antigen. So when that T cell encounters this macrophage, this antigen-presenting cell, then the immune system will be the immune response will be activated. So I'm going to go through this again, but I want to watch a video first. It's a little complicated, but we're going to review it a few times, so it makes a lot more sense.
Antigens are processed differently, depending on whether they originate within or outside the cell. Proteins produced within the cell, such as viruses or self-proteins, are broken down into fragments. Fragments of foreign proteins are antigens. The antigens are then transported into the rough endoplasmic reticulum. The antigens combine with class I MHC molecules.
The combination is then transported to the Golgi apparatus and from there to the plasma membrane. Foreign antigens presented on class 1 MHC molecules stimulate cell destruction by activated T cells. Self-antigens are not recognized by the T cells and do not stimulate cell destruction. A different process occurs if the antigen originates outside the cell. Phagocytes, such as macrophages, ingest foreign particles such as viruses and bacteria by endocytosis. The foreign particles are broken down into fragments within a vesicle. The vesicle containing the foreign fragments fuses with vesicles from the Golgi apparatus containing the class 2 MHCs, and the two structures combine. The MHC class 2 antigen complex is transported to the plasma membrane. The displayed MHC class 2 antigen complex can stimulate other immune system cells to respond to the antigen.
So that video is a little more detailed than you're going to need to know for the quiz. But what it's mentioning is that we actually have part of our self proteins, it's called major histocompatibility class 1 proteins, that connect with part of the processed antigen and then display it on the outside of the cell surface. And this is what T cells will recognize, right? Because this is an infected cell that one specific T cell can recognize and then will kill the cell if it's a cytotoxic T cell. That's how it recognizes that this cell has something bad in it, right? Because it got chopped up and displayed on the surface. That's the only way that this T cell would have known that this target cell is infected. The other way that this can happen is this macrophage engulfs a foreign cell, a foreign pathogen, foreign bacteria, let's say, chops it up, and again displays it on these MHC class 2 molecules. And then this can be recognized by lymphocytes as well. You don't have to know the difference between MHC1 and MHC2 for this class. I want you to understand that when a macrophage engulfs a foreign antigen, it chops it up, and in combination with its own self protein, presents the antigen and its self protein on the cell surface. And then when a T cell that is programmed to recognize that, finds this on the cell surface, it is activated and can form many copies of identical T cells that can then bind to that same antigen that activated the parent cell. So here we'll go through this step by step. Okay, so here we have an infected cell that is displaying part of the antigen that infected it, along with the cell protein on its cell surface. We call this an antigen-presenting cell. Here we have a specific T cell, either a T helper cell or a cytotoxic T cell, that will recognize that antigen fragment on the antigen-presenting cell, and it becomes activated. An activated T cell then divides and forms a clone of identical T cells. A clone, right, means replicating many, many, many times, and it's an identical copy of this cell that got activated by the antigen-presenting cell. The helper T cells will divide into many other helper T cells and also some memory T cells. So these are going to be dormant if ever again this antigen is presented to the body. The cytotoxic T cells will also divide into many, many, many other cytotoxic T cells and also form some memory cells. And then these cloned cells, now that we have tons of activated cytotoxic T cells and tons of activated helper T cells, they can fight the pathogen until it's eliminated. It's basically going to speed up exponentially. We're going to get more and more and more of these specific T cells to recognize any of the infected cells that have antigen on their surface, and will make sure that they're killed. So the way that this will work is that the T cells can specifically punch holes or inject them with a poison to kill any foreign or diseased cell. The helper T cells release cytokines, which promote the activity of B cells, and also will tell cytotoxic T cells to come in and start injecting more poisons into them to kill them. Cytokines also promote phagocytosis by neutrophils and macrophages. And the memory T cells launch a secondary response if the same targeted antigen reappears in the future. So this is the cell-mediated immunity done by T cells. So pause here and see if you can answer this question. So the answer is A. So now we'll talk about antibody-mediated immunity. Antibody-mediated immunity is also known as humoral immunity. And this provides a defense against extracellular pathogens, pathogens that are not inside of cells. They're in the body fluid. These are carried out through B lymphocytes, and these B lymphocytes are often stimulated by the helper T cells that make cytokines. Right. So they provide a defense against extracellular pathogens only, and B cells involve a type of B cell called plasma cells produce antibodies, and these antibodies can then target any extracellular pathogens. Antibodies are defensive proteins. They're also known as immunoglobulins. And antibodies are Y-shaped, like this, and they have these ends of the Ys that bind to specific antigens. They're called the antibody binding regions. So these antigen binding regions each can recognize only one part of an antigen, like a lock and a key. So each antibody only recognizes one part of an antigen. And there are two different binding sites on each antibody. Antibodies can then, once they bind to the foreign antigens, they flag the antigen for destruction by various immune cells. So other immune cells will recognize anytime there's an antibody bound to antigen. And then let's say those cytotoxic T cells will come in and start killing the infected cells. Right? Macrophages will engulf and digest anything flagged with an antigen-antibody complex. So if there's a bacterium floating, a bacterial protein recognized by your antibodies, that antibody will bind to the bacterial protein, and other immune cells will then recognize the antibody and say, uh-oh, something's going on here, and then it will start killing or eating up the bacterium. So the macrophage can just eat up a bacterium that is flagged with an antibody. And then that bacterium will be chopped up and then displayed on the macrophage's surface. So then the T cells will recognize it as an antigen-presenting cell, and then you would have cell-mediated immunity. Antibodies can actually directly neutralize some bacterial toxins. So if there's some kind of bacterial protein in the lymph or in the blood, an antibody can directly bind to it and neutralize it. Right? If it's bound to an antibody, that toxin can't really do much. It's kind of blocked. Right? Its function is blocked, and there's like a huge antibody bound to it. It's stuck. So these antibodies can provide immunity in many different ways. So the antibody-mediated response starts when receptors on the B cell surface, like these antibodies, right? These antibodies are specific to one specific antigen. So we have B cells millions of different B cells that each recognize one specific antigen. And when it does recognize an antigen, right, it binds to it and it gets activated. This B cell will engulf the antigen and display it on the membrane, along with part of the self protein. And remember, this is one of those MHC2 molecules. So the antigen was recognized by the receptor on the B cell. The B cell engulfed the antigen, chopped it up, and displayed it on the surface. Then helper T cells that are programmed for that specific antigen will bind to it and the self protein, and then it will secrete cytokines that will activate the B cell. So this is a multi-step process that you might have to review a few times. So again, step one, the B cell receptor recognizes the antigen. The B cell engulfs the antigen and displays part of it with the self protein on the membrane surface. The helper T cell will recognize this complex and will release cytokines that will activate the B cell. Once the B cell is active, it can divide into plasma cells that can produce antibodies that recognize that specific antigen. These antibodies are just like the receptors on the cell. But now we're going to get a clone of them. We're going to have millions of these cells that could produce antibodies then. I don't know, I'm not sure about millions, so I take that back. Or thousands of these plasma cells that could each make antibodies that can float through our bloodstream and recognize anytime they see this antigen. And these antibodies can move really fast through the body. We also make memory B cells that will remember the antigen and will launch an attack against the antigen should it appear again. So let's pause here and review all of these different cells and make sure we understand each of their functions. So here's a video I want you to watch that is a really good summary of what's going on when you get infected.
This patient has a strep bacterial infection causing a red, sore, pussy throat. The tiny round strep bacteria thrive on throat tissue. The bacteria are coated with hairs called M protein. M protein is what your body recognizes to be foreign. In response, your body will launch an immune attack against it. Your lymphatic network is an important part of the immune system, detecting any infections that arise in your body. The M protein hairs drain into the lymphatic system around this patient's neck. The lymph node is crammed full of B cells. B cells are guardians of your immune system, scanning the body for foreign material. Arriving at the lymph node are M protein hairs from the bacteria. Your body contains billions of B cells. Each B cell is unique and is sensitive to only one kind of foreign infection. By chance, one B cell is sensitive to the M protein hairs from the bacteria. This is the molecular surface of that B cell, magnified 10 million times. Its surface is covered with receptors that recognize M protein. The receptors grab onto the M protein hairs. This triggers an activation signal to travel into the cell. the activated B cell migrates and begins to replicate. In a few days, thousands of copies of the selected cell are made. This is the central idea in Burnett's clonal selection theory, where one cell is selected out of billions of B cells and identical clones have been created from it. Therefore, the cell is clonally selected. This creates an army of B cells that mass produce M protein antibodies and release them into the surrounding fluid.
These are the antibodies.
The antibodies circulate in the blood and travel into tissues. They bind to the M protein hairs on the strep bacteria. This flags them for destruction by the immune system.
And sometimes they don't always look like just the Y, and they form these complexes, by the way.
Other immune cells ingest the antibody-covered bacteria. They destroy the bacteria and eliminate the infection.
Right, so they recognize the antibodies covering the bacteria and then engulf them for destruction. And then they could present the antigens on their cell surface for T cells to then recognize. So to summarize, we said that adaptive immunity has two arms to it, right? It has two branches. We have cell-mediated immunity and antibody-mediated immunity. And it starts when antigen presentation triggers an immune response. So that could be because of a virus or a bacterium. And the cell-mediated immunity will begin when a cell can phagocytose the bacterium and then present part of those antigens on the cell surface. And that will activate T cells. And those activated T cells can then find the pathogens and attack them through chemical toxins or by phagocytosis. So that will result in the destruction of the foreign antigen. The other arm is antibody-mediated immunity. And that is when an activated B cell, which could be activated by helper T cells, gives rise to other cells that can produce antibodies. So plasma cells are B cells that can produce antibodies that can then circulate throughout the cardiovascular system and destroy the antigen. Also, it marks the antigen for destruction by other T cells. So these two branches are very tightly regulated by each other. So there's a lot of communication and feedback between cell-mediated immunity and antibody-mediated immunity.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories. flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
Great. So that was a more detailed but more thorough overview of everything we spoke about so far. So now is a good time to pause before we finally talk about immune responses. So that was a really good overview. I suggest watching that a few times. So now we're going to talk about immune responses. The primary immune response occurs with the first encounter of a pathogen. And this is when T and B cells make clones of themselves to destroy the antigen. So they're doing their thing, they're doing their immune response like we just spoke about. They're also producing memory cells that recognize the same antigen if it ever re-enters the body. But it's taking some time. So it takes about, let's say, 3 weeks after the first exposure to the antigen to make a decent amount of antibodies, so to develop immunity. However, the second time that pathogen invades your body, the response is so much faster and more intense the second time. So look at this level of antibodies the second time around. So this could explain why getting a booster shot of a vaccine really can increase the antibody levels and provide more protection. So there is more than one way for a person to become immune to a pathogen. There's 2 types of immunity. We could say there's active immunity or passive immunity. So if it's active immunity, that's acquired through the use of the person's immune system. The person is directly responsible for becoming immune, and that leads to the development of memory cells. So there's active immunity. There's also passive immunity, which is acquired without the activation of a person's immune system, and no memory is developed. It might sound a little confusing now, but it'll make sense in the next slide. Or in the next 2 slides, I promise. So again, we have active or passive. Active is you're using the person's immune system to produce immunity. Passive is the person's immune system is not being used to provide immunity. In addition, it can be natural or artificially acquired. So you can get immunity naturally or artificially. So I'll talk about naturally acquired active immunity is when a person gets sick and then recovers. It was naturally acquired, a person got sick from a natural exposure to something, and their immune system produced the immune response and then recovered. That would leave antibodies in that person and also memory T and B lymphocytes that would fight later infections. So this is naturally acquired, you got sick, and it's active immunity. The person was actively sick and then leaves antibodies and memory T cells and B cells. Compare that to artificially acquired active immunity, which is a vaccine. So it's artificially acquired. I'm injecting an antigen inside of somebody or something that can trigger an immune response. But that person is still making the immune response, so it's still active immunity. But it's artificially acquired because it wasn't just by getting sick. It was by injection or exposure to something. That would still, since it's active, leaves antibodies and memory T cells and B cells. You can have naturally acquired passive immunity when infants receive maternal antibodies through the placenta and through breast milk. So I should say, so when fetuses will receive antibodies and will be able to have an immune response, but it's passive immunity. It's acquired by not direct exposure. And again, it's naturally acquired because it's coming through the placenta, and this is, or the breast milk, and this is short-term immunity. So this is not producing memory. So that's why it's passive. So this is only short-term immunity for the newborn infant, does not stimulate an immune response. So it's passive immunity. And finally, you can have artificially acquired passive immunity. That's like when you receive an injection of antibodies that were made by somebody else's immune system or some other animal. And again, you're not stimulating your own immune response. So it's still passive immunity. So this will enable short-term immunity without stimulating an immune response. So when you get bit by a poisonous snake, you can inject antibodies against that toxin if you want to save yourself. But if you get bit by the same snake later on, that response won't be any faster. So it's passive immunity, and it was artificially acquired because you didn't make those antibodies. They were actually made by an animal, or they were synthetically made. That's called an antiserum. So that's used in emergency situations when the pathogen acts very severely. So we heard about antibody infusions for COVID. For like really severe cases, you give them antibodies that were produced from somebody else who already had COVID. So that's artificially acquired, and it's passive immunity. So if that person were to get COVID again, their response wouldn't be any faster. For that, you would need to get the COVID vaccine, which would be artificially acquired active immunity. I'm injecting the instructions on how to make a part of the COVID virus. So I'm making the COVID virus inside of my body when I get the vaccine. Just a part of the COVID virus, just a part of the protein. So my body will then produce B cells, a clone of B cells that recognize that part of the bacteria, the viral protein. So when I do get exposed to COVID, the actual virus, I already have tons of actively acquired antibodies. My body already has an immune response and already has memory T and B cells against COVID as if it were already in my body naturally. So when I do get it in the real world, I hopefully won't get sick. Or if I do get sick, I won't die from it. I won't have to go to a hospital. So vaccines enable artificially acquired active immunity. And these have produced enormously successful public health campaigns. We've eliminated or nearly eliminated polio, smallpox, measles, diphtheria, tetanus, and many other infections. Vaccines also enable healthcare workers or those traveling to foreign countries to be protected against some viruses or bacteria that might be prevalent in other places. However, as we know, modern fear or refusal of vaccines is causing many of these diseases to return and many diseases like COVID to continue to spread. And if you doubt the benefit of a vaccine, just Google the symptoms of any of these diseases, and I assure you that the disease is a lot worse than the vaccine or any threat of what the vaccine could potentially do. This is a great chart to look at that looks at the decrease in the cases of all these different infections. So we see 100% decrease in diphtheria. We went from 21,000 cases of deaths every year to 0. From the flu, hepatitis, measles, polio, smallpox, etc. So pause here and answer this question now. So how do vaccines work? And the best answer is A. It is artificially acquired active immunity produced by exposure to altered antigens. Right, so it's not the actual antigen, It's some kind of altered antigen that's synthetically made, or it can be an antigen. Sometimes they could take a virus and weaken it. So it's not a live virus. So when it does get inside of you, it can't really make you sick, but it could initiate an active immune response. So I'm going to stop there. We're at about an hour and a half. I'd like you to review the remaining slides in the PowerPoint. There's nothing very complicated about them. And please just refer to the learning map as to what you must know for the upcoming quiz. So I'd like you to review a little bit about just organ transplant and clearly how that might be a problem for the immune system as recognizing a foreign antigen. It's recognizing the organ as a foreign antigen. And a little bit about some of the disorders. And then you're going to present some to me during the next class. So I hope you learned a lot about the lymphatic system and the immune system. And I will see you next time, where we will discuss the respiratory system and the digestive system. So thank you for coming, and have a great day.